In this chapter, we will provide a brief introduction to functional magnetic resonance imaging. Functional magnetic resonance imaging, or fMRI, is an imaging technique often used in neuroscience research to identify the metabolically active parts of the brain. fMRI was designed to extend the capacity of regular magnetic resonance imaging, or MRI, which is an imaging technique that is typically used to create detailed static images of the organs and tissues in our bodies. Unlike MRI, fMRI is used to capture functional changes in the brain caused by neuronal activity. This is made possible by using magnetic pulse sequences that help reveal which brain regions have high metabolic demand, presumably because the neurons there are firing lots of action potentials. This technique can detect changes on the scale of seconds, making it a great way to identify rapid changes in brain activity. This method is particularly useful for researchers who are trying to answer questions about the parts of the brain that are active when a person is completing a particular task, such as reading, writing, or making decisions. In order to identify metabolically active regions of the brain, fMRI analysis involves using the blood oxygen level dependent contrast, or BOLD contrast, to compare periods of activity with periods of rest. This method uses magnetic fields to track the changes in consumption of oxygen in a person's brain. Specifically, this method compares the levels of oxyhemoglobin, which is the oxygenated form of hemoglobin in the blood, to deoxyhemoglobin, or hemoglobin that has donated its oxygen. When particular brain regions are active, they receive more blood, and this blood donates more of its oxygen. Since active neurons demand more oxygen compared to non-active neurons, fMRI detects changes in blood flow to reveal the regions of the brain that are more active under various circumstances. The unit of an fMRI image is referred to as a voxel, or a volumetric pixel. Each voxel has an intensity value, which reflects how active it was. The voxel has a 3D location in the brain and a timestamp, so we know when the activity occurred. Brain activation detected from fMRI is often represented by color-coding metabolically active voxels across the brain or the specific brain region of interest. Activity in those areas can then be analyzed in response to the task that was performed while the participant was in the scanner. To detect a change in activity, we need at least two conditions to compare. Bold signals in fMRI are therefore usually compared between what is seen during a task to some kind of control condition, such as just being at rest. We often represent this comparison like the figure shown here. Clearly, there are many regions in the brain that are active during the task, but the regions colored in red are the ones that are more active during the experimental condition of interest, as opposed to the control or baseline condition. Contemporary analysis techniques also make it possible to analyze brain responses when participants are not doing a task, such as passively watching a movie, or even while they are sleeping. These techniques have found that certain regions in the brain, including regions such as the posterior cingulate cortex and the ventral anterior cingulate cortex, are more active when people are at rest compared to when they are actively performing a task.
Before being recruited for the experiment, participants are screened to ensure that they can be scanned safely. Participants are also asked to remove all metal, including things like jewelry, glasses, and cell phones. This is because the scanner is essentially a large magnet, and these objects can cause damage to the equipment and, more critically, to the participant, as the large magnet can forcefully pull these objects towards it. During an fMRI scan, the participant is asked to lay face up on a flat table that slides inside of a large cylindrical scanner, referred to as the bore of the magnet. Then they're given instructions regarding what to do while inside the scanner. Sometimes screens are used to project images or movies to participants inside the scanner, and the participants are asked to make responses to what they see by clicking a button with their right or left hand. Cushions are used to make participants comfortable as they lie in the scanner and also to minimize movements while the data is acquired, as motion can severely distort the quality of the data that is collected. While the participant is in the scanner, researchers in an adjacent room are able to see and record images of the participant's brain as they perform the specific task. A typical fMRI scanning session lasts about an hour. In our work, we study regions within the occipitotemporal cortex that respond preferentially to certain categories of visual objects. Currently, our work is focused on a region that prefers word-like character strings to all other forms of visual input, which is referred to as the visual word form area. While showing participants words can reliably elicit responses within this region, we are exploring whether performing a language task while viewing these stimuli systematically affects these responses. Our results show that both what is perceived and what a person is doing influences activity within this region. We also use fMRI data to study responses across the whole brain. For example, to determine which brain regions co-vary in their temporal responses in a similar way in different parts of the experiment. Many researchers choose fMRI over other brain imaging techniques because it is non-invasive, meaning it does not require the use of things like injections, surgery, or the ingesting of substances. Additionally, fMRI has better spatial resolution than other non-invasive techniques, such as electroencephalography and magnetoencephalography, meaning that it does a good job discriminating between nearby locations. fMRI data also allows researchers to collect data from the entire brain and to identify regions that they're interested in studying after the data is collected. However, fMRI is not a perfect method. For example, compared to electrical techniques like EEG, it does not have great temporal resolution, which is the smallest time period of neuronal activity reliably separated out by a technique. That is because the fMRI signal represents an increase in metabolic demands following neuronal activity, which is an indirect measure of neuronal activity. Additionally, fMRI data is susceptible to distortion from head motion that may occur while the participant is inside of the scanner, which can lead to artificial intensity changes of the fMRI signal. There are also some small risks associated with fMRI for the participant, including claustrophobia, exposure to loud, high-pitched noises, and possible nerve tingling. All of these effects can be mitigated by screening participants before starting an fMRI experiment. In general, however, fMRI is considered a safe brain imaging technique that is extremely useful for mapping brain activity.